In the next 50 minutes or so, I will be talking about alpha-predicted models for cryo-EM. So it's more on a practical points of view, so aspects. So I work in the Phenix group. So there will be here and there some practical tips or screenshots basically of the Phenix software, how you can do certain things. And I will share my slides. I know I got an email or with a link to upload. So I usually do this after the talk because I like to do some last minute changes. So no need to frantically take any screenshots. The slides will be made available. Okay, so let's get started. So here's an outline of the presentation. I will talk first a little bit about AlphaFold predictions. Some reminders, I guess most of you know this already. And then I give some suggestions how you can use predicted models specifically in cryo-EM. And so I will focus on docking. I will talk a little bit about model completion. And another aspect is reference model restraints in Phenix. And finally, I will also suggest an automated workflow that will iterate between prediction and model building. So let's start with predicted models with AlphaFold. As most of you know, now with AlphaFold, it's possible, given a sequence and a multiple sequence alignment, to get quite accurate 3D predictions of proteins. And the nice thing here is that you not only get a structure, but you get something called confidence measures. And these confidence measures are very useful for the downstream use of these predicted structures. And so here I want to just remind what kind of confidence measures there are. So the first one is the PLDDT, that stands for Predicted Local Distance Difference Test. And this value identifies where errors are more likely. This is a per residue confidence measure, so each residue has a number. And this number scales from 0 to 100. And if this PLDDT is larger than 90, then we typically say that this model or this residue is predicted with high accuracy. And so this as a matter of fact, the low sequence coverage can contribute to low PLDD score. This relationship it's not a one-on-one relationship, but here we see an example where it kind of relates with each other. So here we have a protein that is about 130 residues long. So here on the x-axis, you have the residue number. In this plot on the left, you have the sequence coverage, shown here by this black line. And sequence coverage is quite okay between residues 0 to about 100, and then it starts to drop from 100 to 130. And here on the right plot, we have the PLDDT, and we see a kind of similar behavior. Between 0 and 100, we have a high PLDDT here in the 90 to 100 range. And then at around residue number 100, we have this drop. And let's look how this translates in the prediction. So here we have the corresponding prediction in green. That's the AlphaFold model. And in lavender, you have the deposited structure. It's 7MJS. That's the PDB code for this model. And we have quite good correspondence between the green prediction and the deposited structure in this region here. And that's residues 1 to 100, where we had high sequence coverage and high confidence. Whereas here in this loop region, that's residues 100 to 120, we have low sequence coverage, low confidence, and also low accuracy. And so here is a handy chart that relates the PLDDT to prediction errors. So this is based on a study by Tom Terwilliger. He looked at, I think, 200 experimental SAD, so X-ray structures, and compared the prediction with the refined structures, so redetermined structures. And so if PLDDT is in the high region, so larger than 90, median prediction error is about 0.6. and percentage with residues over 2 angstrom is about 10%. Let me just use the little screen, not annotation. I want to have the spotlight. Nope. Sorry, I want you to be able to see my laser pointer. Can you see that little red dot?
Yes.
Okay, yeah, that's much easier. All right, if the PLDDT is between 80 and 90, so a little bit less good, we can see median prediction error increases, and also we have now more residues with larger errors. And correspondingly, this goes larger if we have lower PLDDT. Now, PLDDT of about 70 is typically used as a cutoff if we want to say something has been predicted correctly or incorrectly or not good enough. And here we see very well why. If PLDDT is less than 70, we have a quite high prediction error of 3.5 angstrom, and almost 80% of residues have an error over 2 angstrom. So it's very handy when we have a predicted model to color to look at the prediction in a viewer and color the residues by PLDDT. So here's an example of an RNA helicase. So this is an AlphaFold prediction. If the color is in the blue range, we have PLDDT larger than 90. If it's green, it's between 80 and 90. And here in the lower right, you have again this little chart so you can see to what coordinate error that corresponds to. Now, this is the first confidence measure that we get with AlphaFold predictions. That's the PLDDT, and there's a second one, and that's called the predicted aligned error, or PAE. So this confidence measure essentially informs us about the certainty of relative positions between two residues. And in the end, this will basically identify accurately predicted domains. So let's look at this plot here in the bottom. So this is again this RNA helicase from the previous slide. And this is about 370 residues long. So on the x-axis in this plot, we have residue I. On the y-axis, we have residue J. And in blue, we have the uncertainty. This plot is colored blue if the uncertainty in the relative positions between residue I and J is less than 5 angstrom. So let's, for example, look at residue number 100. So now if we go all the way up here, we see that from about 0 to about 210, so this region is all dark blue. So between the two residues, the relationship, the uncertainty is low. That means relative position is determined with high accuracy. Now, if we go to higher residues, so from 210 to the C terminal, here we have higher uncertainty. So we are less certain how the relative position is. So in other words, if we see here in this plot these squares, these represent domains in which the relative position of the residues is quite certain. So here we have two squares along this diagonal. And so that tells us that most likely the relative position in the AlphaFold prediction is not so clear. So if we want to use this AlphaFold prediction, let's say for docking in cryo-EM, we probably would like to cut this full prediction into two smaller parts and try to dock these two components separately. And that's exactly the same thing that is done in crystallography as well. Because we don't know how these two components or these two domains are arranged with respect to each other. So this is about the PAE. And then there's one more thing that we need to be aware of, and that is that the PLDDT value is actually stored in the B factor column. And so let me just illustrate why this may be a problem, not necessarily a problem, but it's something we should be aware of. So again, here is an AlphaFold prediction. Blue means high PLDDT. If we go towards the yellow and orange, that goes to lower PLDDT. So high PLDDT means high confidence. Low value of the PLDDT is low confidence, so position is more uncertain. The PLDDT value in the model file, so this is really a practical issue here, the PLDDT value is stored in a column that is usually reserved for something called the B factor. And the B factor is a description of the disorder in a macromolecular model. And the relationship between PLDDT and B factor is exactly inverse. So if we have a high B factor in a macromolecular model, then this typically means that this residue is disordered, meaning its position is probably more uncertain. If the B factor is low, it's more ordered. So relationship here is exactly inverse. So here I switched the color scheme. If we would interpret a predicted model in terms of its B factors, everything that is blue here would be red, and red here means less certain. So if we want to use these predicted models downstream, we need to convert these PLDDT values to pseudo B factors. So for example, B factors may be used for weighting in docking, meaning that residues with high B factors are downweighted. So if we just use the PLDDT values as is, we may downweight the wrong kind of residues. And here, just for completion, I show you how you can do that. There are formulas that allow you to do that relate the PLDDT to the B factor. But in reality, you don't need to know the exact values here because this is done in programs automatically. And the reference where this is described, you can see here in the bottom. So to summarize, these AlphaFold predictions, they are great hypotheses because they can be really awesome. So here in this picture on the left, you see an experimental map. I don't remember if it's crystallography or cryo-EM, but in pink, you see the AlphaFold prediction. So this is just a prediction put into that map without any refinement or anything. And you can see that there's really a striking correspondence, right? The side chains are almost exactly everywhere in the map. But sometimes they can be just wrong. So here's another example. Again, in pink, we have the AlphaFold prediction. We have the map for this macromolecule. And here doesn't look quite right. Maybe here some correspondence, but that doesn't look like it fits the map. And sometimes it's in between. There can be some distortions. So especially let's look maybe at this helix here, right? This clearly is a density that has helical shape. We see that the protein chain has also a helical shape, but it's a little bit displaced. And I think also the helix in density is a little bit more less squished in. So the model has slightly different geometry. So there is a distortion. It's not quite right, but it's also not completely wrong. And for all three cases here, the PLDDT values were actually high. So still, even if with these very useful confidence measures, they're not necessarily reflect what is done in the actual structure that is compared with experimental data. So how can we use these predictions? So there are already multiple ways how we can incorporate these predictions into typical cryo-EM workflow. And so here, just in a nutshell, I'm pretty sure you're all familiar to this. The cryo-EM workflow is typically starting with assessment of the data quality. So we look how good is our map. If we can improve the map by sharpening or density modification. When we have the best possible map, we would dock a model into that map. maybe we have to rebuild a little bit, and then we can go into that cycle of refinement and validation that ultimately will lead to the final structure that we can deposit and interpret for to answer our biological questions. So where can we use these predicted models? I think obvious places would be here: docking, rebuilding, and refinement. So docking, obviously, we would dock a predicted model. We can also use AlphaFold models as hypotheses for model rebuilding, so build missing parts. And in refinement, real space refinement, we can use it as reference model restraints. And so I will go now through each of these examples and suggest some applications. So let's start with docking. So using a predicted model for cryo-EM docking. So typically cryo-EM maps have quite low resolution, and so it's very challenging to build a model ab initio into such maps. So very often the workflow is to dock a pre-existing model into the map, and that can be a homology structure, that can be a known crystal structure. And if this is not available, we can use also an AlphaFold or a predicted model. So we use a model that we assume looks like the sample. We have a map. We find the translation and orientation, or the program tries to figure that out, and we end up with a docked model. So let's just walk through one example. So here we have model 7BRM. That's one of the models that we have in our tutorial data that we ship with the Phenix software. The map has a resolution of 3.6 angstrom. And let's say we want to solve the structure. So we don't have the model just yet. So we just have a map and the sequence. And so here's the sequence of this compound, and here is the cryo-EM map. So first thing to do, we have our sequence, put it into AlphaFold and get a model. And just to wake you up, what do people think? Is this a good prediction or not? So you can either use the chat, or you can use like thumbs up or reaction. What do people think? I try to open the chat, always challenging with the presenter view, especially if that little, here we go. Okay, someone says no, weak prediction. Okay, why? Why do you think this is not a good prediction? Maybe you can unmute, right? It's no problem to. Everybody can unmute, or is there any restriction, Jake?
They should be able to.
Okay, yeah, I have some, yeah. For example, Pradeep, you said in the chat, this is not a good prediction. Would you like to explain
yeah.
why you think?
Yeah, definitely. Because the conformation looks quite different to that of what you showed from the cryo-EM map.
Okay, let's try if I can get back. Here we go. Okay, well, yeah, that can be a different, maybe I don't have the right orientation. I think in these pictures, I don't have the same orientation. So it's maybe a little, so see, here is this beta sheet. If we go back here, yeah, it's a little bit rotated back. But we have, yeah, right. Maybe I should have shown the map in a different orientation. So maybe that was a bit misleading. We have someone else who thinks it's a weak prediction. Maybe, yeah, Daniel Rubin, would you like to comment? Or you can also put it in the chat if you cannot speak. Okay. All right. So I actually think it's not that bad, at least for alpha. Oh, sorry, it's good prediction. It's similar to the cryo map. Seems like they resemble. Okay. Well, we talked a lot about these confidence measures. So just looking at the predicted structure is, I think, not enough. So we have to look at what the PLDDT, and well, here the PAE is not so informative because it's actually just one domain. But before that, generally, this is probably not such a hopeless case because we can see helices, we can see sheets, while there are some weird loops here and there, but generally this looks like what we expect a protein would look like. So here I show you okay, this is an example of I'd say, more weak prediction. See that there are some parts where we see sheets, helices, but there are long, long stretches of loops just hanging in space. So this is what our collaborators from the Richardson lab call, oh yeah, no, I forgot the name, barbed wire. Yeah, this is just a line of main chain that hangs in space. So here visibly, this looks kind of odd. And the prediction we have here doesn't have this kind of pathology or this kind of oddity. But still, we have to look at the confidence measures. So let's have a look at the PLDDT. So now I colored. It's the same view, the same protein. I just changed the color. So each residue is colored by its PLDDT. Blue is high PLDDT, so high confidence. Yellow, orange, red is low confidence. So first of all, we don't see any red. Most of it is yellow, so greenish, bluish. So at least AlphaFold itself is quite confident with this prediction. So if we want to use this predicted model now for docking, we have to process this model. We want to remove everything that is predicted with low confidence, so these yellowish regions. And we don't have to do this by hand. We have a program that will do that. And that is called Process Predicted Model. Here's a screenshot. If you're familiar to the Phenix GUI, you go to AlphaFold, and there are several options, and one of them is Process Predicted Model. This can do three things, the three things that I mentioned. It will remove low confidence residues. Typical cutoff value is 70, but you can you can change that according to what you need to do. 70 is usually a good default. We replace the B factor field from PLDDT to these pseudo B factors. And if the PAE plot suggests different domains, you can supply the PAE matrix, and then it will try and interpret this and cut the protein into different domains. So all this done automatically, and we get this model here. So sorry, if I go back just right, this is the complete AlphaFold model. If we get rid of some of these yellow regions, we end up with this model here. So here where we have these lines, these are the gaps. So here we have this little helix flying around in space. So probably we don't want to use that downstream because this is typically too small to be used in these docking programs. So now here's the map, here's the model, and now we want to dock. And let me just summarize which options you have with Phenix tools to dock this model, this predicted model, into the map. So our older version of docking is called Dock in Map. You find it in the docking and model building category in the Phenix GUI. And this would just do basically a brute force search, trying to find the translation and orientation of your model and tries to fit it into the map. So here's the name of the command line tool for those of you who are using command line, but it is available in the GUI. So this tool has been developed by Tom Terwilliger. And then a newer tool that is called EM placement. So this tool will also do docking. And it has been developed by Randy Reed's group in Cambridge. For those of you who do crystallography, Randy Reed is the developer of the phasing of the molecular replacement software Phaser. And so in Phaser, they use maximum likelihood approach, and they use the same approach now for cryo-EM docking. So this program uses likelihood scores to dock a model into a map. The big advantage of this likelihood approaches is that you can take into account errors of the map, errors of the model, and also incompleteness of the model, and you can still get scores that tell if this particular orientation of the molecule in the map is better than a random orientation. And this works also at low resolution, I believe. So I have the two papers listed here that describe this method. Probably the lowest resolution described in this paper is 8.5, but I think it goes even to lower resolutions than that. So this is a very powerful way of docking. And here you see two screenshots with examples. And you can see that the maps are very noisy, and it's still possible to find this docking solution here. Now, this is called EM placement. It's relatively new. And we have also, there exists a so-called local version. It's called local EM placement. And this is not used in a GUI, but this is can be used in Chimera X. So Chimera X has now a few Phenix tools available, and one of them is this likelihood-based docking. The idea here is that it will not try to do docking in the full map, but in a local region that you can visually choose in the Chimera X viewer. So you can select the region into which the model should be docked. And so I have a video here.
Hey, Dorothy, we have a couple of questions in the chat if you want to take them
Okay,
now.
sorry.
No,
The
it's okay.
chat
It's
is
hard
in a
for you to
different
see.
window. So yeah,
Yeah.
we can have a break and go over these questions. If you don't have a cryo map, is the two prediction metrics enough for deciding whether or not it's a good structure? Oh yeah, that's a good question, right? The problem is, yeah, if there is no experimental data, be it X-ray or let's say any experimental data is very difficult to say if this is true or not. It may be, because there are many cases where the AlphaFold prediction is very similar to the experimental data, but in some cases it's not. So very hard to say. But, and then the second, any other things to refer to for assessing prediction? I'd say these are the two. Well, I would say maybe, yeah, if the directly related to the prediction, that's probably the two big metrics that you have. Okay, the next question, Alejandro, is docking and cryo-EM similar to molecular replacement in cryo-EM? So I am not so sure if I didn't really hear molecular replacement for cryo-EM. Molecular replacement is used in crystallography. So maybe Alejandro, if you could expand a little bit, you can unmute yourself and maybe say more.
Yeah,
or you can put it in the chat. Yeah, okay.
Yeah, thank you. Yeah, my question was more related because when you were explaining about the docking process, I'm a crystallographer by training. So when you were talking about docking, it immediately resembles for me in my memory about the molecular replacement process that we do in crystallography. I don't know if there's, yeah, and you spoke about the likelihood. So I think there's some similarities there in those methods, right?
Yeah, definitely. That's why it was Randy Reed who wrote this phaser software. They thought that this is a very similar problem, right? Finding translation, rotation of something, and also having these noisy maps that you need to find a score to tell you if that particular pose is actually correct. So yes, that is similar. I think that was just a typo. So yes, docking in cryo-EM here in this in-place local tool is similar to molecular replacement approach in crystallography.
Cool, thank you.
Okay, are there any other questions? All right, then I try to start that video. Maybe I do have to remove that little laser pointer. Oh. Okay, so I do need to get rid of my little laser pointer. If I manage to do that, it's really hard to. Oh. Yeah, let me go here. Okay, here we go. Sorry for that. It's the little laser pointer that throws me a little off. So here is how you can do docking in Chimera X. It's this local version of this emplacement, this maximum likelihood docking approach. So here we go a little bit through that video. We have this little protein here. This is a subunit. It's an X-ray model. And we want to put it into this density of this respiratory complex. Density is very, very noisy. Now, the nominal resolution of the entire map is not too bad. It's 3.8 angstrom, but this density is only 9 to 11 angstrom. Now, in Chimera X, in practice, how you then do this, there is this yellow sphere that you can move around. So I stopped the video here. You can move that yellow sphere around and define the region where you want to place this model. The size of the sphere, as you can see here, is related to the size of the model. So if you have something bigger here, that sphere will be bigger. So you can search for something that will encompass your model. So you can move it around until you're happy, right? So here, moving. And then you start the process. Oops. Do you start the process and it will dock? So here it's instantaneous. In reality, it will take about a minute, I think, for this example to be docked. So here it is. You get a certain LLG value. So that LLG value is a score for how well this model fits the map. And now I'm showing a little bit comparing with the deposited structure. Yeah, so here in green, you have the deposited model, in pink or violet, the model that I just docked. And now again, we see the cryo-EM map. And that cryo-EM map is really noisy, right? So if I had to move the model manually into that density, I don't think I would do as good a job as the docking tool. So it works really well even in these noisy maps. So these are the three options that you have in Phenix now for docking. So you can use docking map, this maximum likelihood approach, and you have this visual approach here via Chimera X. So now let's go back to our little example for how to use this AlphaFold predicted model in Phenix. So you can dock the model by any of these methods that I just showed you. And now in blue, you have the docked AlphaFold model that has been processed, so it's not complete, in the map. Doesn't look too bad, but we can identify just visually, just by looking at the screenshot, there are a few regions where there's some clear deviations. So here this better sheet kind of bends out of the density. Here I have this terminal region that hangs here around in the center between these two regions here, but here's some clear density that shows where this main chain should go. And finally, here on the right, we have a loop that is not too far from the density, but I think this is too far for something like real space refinement to fix it. So this needs some either manual rebuilding or some automated rebuilding to put that into the correct density. Some parts therefore don't fit into the map. And here I have, so I've shown you the map. Here I show you the comparison between the AlphaFold model and the deposited structure that fits the map in golden. And so here again, we just see, it's another representation of these parts that don't quite fit. So next thing that we can do in the real world, when we have a case like that, when the AlphaFold model deviates here and there, we have to rebuild. What you could do if these parts are small, like this loop here or this N-terminal region that's not quite right, we have a tool called fit loops. It's not only for loops, you can use it in little helix stretches or in sheets as well. The idea is just, it's about 10, 15 residues that are not quite right. You can ask it specifically to rebuild this part. And then you get this violet model that fits quite well over the deposited structure. So let's move this. Are we having a question? How do you retain where to dock your AlphaFold predicted model? I'm not exactly sure what the question is. I think I'm thrown off with the retain. Do you mean how to save where it is in the Chimera X viewer? Daniel, Ruben? Is that the question? Or maybe in the chat?
Daniel, maybe you could clarify in the chat. Yes, retain.
Yes, retain. Yeah, I'm not a native speaker, so so I'm not so sure. Can you find a different word? Because I'm not exactly sure what the question is. Or somebody else.
I'm sorry, I'm not sure I understand either.
Yeah, okay. Well, if it is saving, if we're talking about the Phenix Chimera X, in Chimera X, you can then just save the new position of the model. You go to File and you ask it to save that model that has been placed. Now, if you are in the Phenix GUI, an output of all these docking procedures is the docked model, so that will fit the map. Oh, you pinpoint a location of Chimera where to dock your model. How do you pick the location? Yeah, well, you would have, in this case, if you do it visually, you would already have an idea where to place it. And usually, I mean, in the scenario where you know what your sample is, right, then you have your map and you have an idea where that particular component should be. So you would look at your map and think about where your model belongs to. Now with a big, let's say you have a big protein with many different domains and you don't know, I guess then where each should be, because this is the first instance of structure that you solve, I guess then you would have to, with trial and error, you would have to check different regions. Okay, so I think I caught up with the questions, so we can go further. Okay, so in the real world, this would be now the moment where we go to the next program, real space refinement, and try to fix all the smaller things. So this is for docking. Now we go to the second application that is potentially useful with these AlphaFold models. That would be to use a predicted model to complete your structure. So here's just an example of a structure that has some gaps. So here you can see where these lines are. We have some gaps. You have the number of residues that are missing. And we could use a predicted model as hypotheses for these missing parts. So here's an example. It is from crystallography, but it highlights what could be done. So here is a complex, a repressor DNA complex that has been solved with something called experimental phasing. For those of you who know crystallography, So the structure has then been built ab initio. And then this model has been refined against 3-point angstrom native data. And this model has some gaps. So here between residues 118, 128, and 139 and 137. Now, at this stage, the agreement between model and data, that's measured in crystallography by something called R and R-free, there were 27 and 29%. This value, as opposed to correlation coefficients that you may be used in cryo-EM, these are better if they're low. So this is quite high. Not shocking at this resolution, but here we see now the density superposed to this model. And indeed, it looks like there's not so clear density between these gaps. But let's now superpose the AlphaFold prediction. And okay, it looks like here is a better sheet that is for these gaps. So why don't we use this prediction as a hypothesis? If we overlay this again to the density, that looks actually not too bad. What was throwing us off maybe here was that the residues here didn't point into the direction where maybe the main chain would actually go. They're pointing here as if they're going somewhere into the void. So if we include this better sheet in the structure, INR3 drops, so gets better, because here we are in crystallography, to 21 and 24%. So in this particular case, filling the gaps with the confirmation predicted by AlphaFold was a good hypothesis. So it's not always, it's not necessarily that this works, but if there are some gaps, why not trying to fill at least part of this missing gap with AlphaFold prediction? So this is the second application for predicted models. And the third one is reference model restraints. So before talking about reference model restraints in particular, I just want to remind what restraints in the context of crystallography, in the context of macromolecular refinement are. So restraints is a source of a priori knowledge, something that we know of our macromolecular structures. They increase the number of observations, and technically, they modify the target function used in refinement by creating relationships between independent parameters. So here's an example for bond lengths. So a very typical type of restraint is stereochemical restraints, and bond length is 1. So imagine this little molecule here, very simple molecule. Now the coordinates of these 2 carbon atoms here, they are independent, right? They don't know about each other. However, if we introduce restraints, a bond restraint between these 2 carbon atoms, the restraint part in the refinement procedure will keep the distance within a certain target value. Let's say we have a good idea that normally in this compound, the distance between the atoms is a certain value, so 1.3 angstrom. And then we can introduce this restraint, and it will keep the distance between the atoms around that value. It will not force it to be exactly, but within a certain deviation. And so if the distance becomes too big or too small, there will be a penalty in refinement. So this is the idea generally of restraints. And there are all sorts of restraints. As I said, stereochemistry, that's used universally, I'd say. And then there are some more derived types of restraints. And one of them is these reference model restraints. The concept here is to use a related model to generate a set of torsion restraints. And the idea is to restrain each torsion angle in your working model, so that's the model you want to refine, to the corresponding angle in a reference. And the way this is implemented in Phenix while we restrain these torsions, it still will allow for structural differences. And I will show in the next slide what that means. So this kind of reference model restraints is useful at low resolution, so typically worse than 3 angstrom. So a good candidate for many cryo-EM structures or maps. And if there's no high resolution homolog available from crystallography or another cryo-EM study, then you could also use a AlphaFold model as a reference model, because these models typically have good geometry. And this good geometry may serve as, let's say, an idealized version of your working model. So here I'm showing you what I mean with this allowing for distant differences. So there is a parameter in these reference model restraints, which is the limit parameter. So if the dihedral is, or the torsion is, deviates by more than 15 degrees, we say these two orientations or these two dihedrals are genuinely different, so we don't restrain them. So here are 2 examples. Here you have a side chain. They are not quite the same, but very, very close. So in let's say in blue, we have the reference model. In red, you have your working model. If they're very similar, it will say that these 2 torsions should be identical in the reference and in the working model, in the model that you want to refine. Now here we have a searing side chain that is off by almost 180 degrees. And so this is obviously larger than this limit. And so here it is considered that, well, maybe there is a genuine difference between the 2 models, so we're not restraining the torsion. So all this is done automatically in the reference model restraint algorithm. You don't have to look at each of these side chains and give it make a decision. This is done under the hood. But this is just to illustrate that what it means that there may be some differences are allowed. So again, here's an example from crystallography, because this reference model restraints is not a new concept that is around for a while already. So in black, you have a low resolution structure at 3.0 angstrom. And in blue is the reference model at 2.3. It's an aminotransferase, in case you wonder what this is. So here we have just the main chain. And actually that looks very similar, right? There's very, very good correspondence, looks almost basically the same, right? There's not much difference. However, if we now look also at the side chains, so now they're not superposed anymore, but next to each other. Again, on the left, we have the reference. On the right, we have the working model at low resolution. And now you can see that some side chains are shown here in yellow. They are shown if they have rotamer outliers. So if the side chains have torsions that are unusual, that are not found in many protein structures. So a few rotamer outliers are okay. So in that model on the left, I'd say that it's not very shocking to have a few outliers. Few outliers is totally fine, that is normal. There may be data that support these outliers. It's not that they're not allowed. But on the right, in this low resolution model, there are way too many. So a few are expected, but this is too much. And it's more likely that these outliers exist because this is a low resolution model. So not the side chain density is not that clear. And as a consequence, these side chains kind of just flop around without much experimental data supporting their modeled conformation. So in this case, it would be useful to impose these side chain conformations from the reference structure. And then we can reduce the amount of outliers. So in case the side chains are already very different, let's say outlier in the working model, and it's a good, it's not an outlier in the reference model, you could say, well, but if this is larger than this limit, will it not keep them separate? So as a first step, here these outliers will be put back into more common conformations, and then this comparison is actually done. So this is how we can get rid of all these unusual amount of outliers in the low resolution structure. And so as a reference model, you could use an AlphaFold model. So here is how to do that in case you are not familiar to it. This is a screenshot of the Phenix GUI. In your refinement, Phenix Real Space Refine, you would supply two model files. One is the model file you want to refine, and then there is a second model file where you have to tell the program this is a reference model. And then if you go to this refinement settings tab, where you give all the parameterizations of your refinement run, you would check this reference model restrained button, checkbox. So these are the three suggestions where you can use your prediction. And finally, I want to go to a little bit different way of thinking how you can use these AlphaFold predictions. And here the basic idea is to use the working model to get a new prediction. And so here's how that would work. Because the new prediction may be better than your working model. So for example, you have an AlphaFold prediction, you dock it, you refine it, you get a better model, you get this one. And then you can use this working model as a template in AlphaFold prediction and get a new prediction that's maybe better. So here we go through this one by one. It's actually this little example shown here in the bottom, but we look at it a bit more in step by step. So this is again from one of our tutorial datasets, 7MGS. It's a single particle cryom structure. Here you have the complete name. The map resolution is 3.03, basically 3 angstrom resolution. And it has these 3 domains, X in red, blue, we have chain L, and here we have chain H. And here we are focusing here on chain H. So in this little chain here, we have a loop that interacts with other chains that is not correctly predicted, right? This little loop here. So let's see in more detail. We have the sequence. Again, we can get an AlphaFold prediction. Doesn't look too shocking, but if we color it by PLDDT, very clear that this loop is much less confident than the rest. So this loop is predicted with low confidence. So again, we go through process predicted model to remove this low confidence residues. And then we dock this model into the experimental map like this. Okay, so that fits quite okay into this density. That looks good. The contour is maybe not very telling, but this fits the density. If we superpose now again the AlphaFold prediction, we see that this poorly predicted loop just hangs here in the void. There's no density here. Does not fit into the map. This blue density, I just colored it here so we see it a little bit better in this screenshot. This is where this loop should be. So if we rebuild this, can be done with any of these tools such as Phenix Fit Loops, we can build this missing part back into the quite clear density. So we have now a much more complete model. So this is a quite good model. So we could think, okay, maybe now I'm done. I can move to Phenix Real Space Refine and move on. But we can actually go back, circle back to AlphaFold prediction. So this time we will do a prediction where we will supply this sequence, but in addition, we will also supply a so-called template. So we can supply like an example, an hypothesis for this prediction into the AlphaFold program. In this template, in our working model, we have the good loop. And if we're lucky enough, this round of AlphaFold prediction will result in a model that also has a good loop. So sequence plus the template will result in a better prediction. So what we've just achieved by doing this is we indirectly supply information of the experimental data into AlphaFold. I'm saying indirectly, right? AlphaFold doesn't have any way right now to, let's say we give it a map and it will use the information from the map in the prediction. It's indirectly because we give it in the form of this model that fits the density. But yeah, it is quite astonishing then that it can make a better prediction. And so do we want to circle back is that this AlphaFold prediction then maybe has also in some other regions better geometry than our working model. And so we would, so here's just a overlay of the prediction using the sequence alone in violet, where we have that bad loop, and in green, the prediction with the sequence and the template. So this good, well-predicted part still is good, but now we have that good loop that fits the density. And so here is we can do this several times, right? So we improve that model, do a new prediction, improve the model using the experimental data. And if we then compare the final model from this iterative procedure to the deposited model, that looks very, very similar. So here you have the overlay of the main chain. And here you have also comparison of the side chains. So even the side chains, there are some deviations, clearly, but many side chains also are very, very similar. So this would be then a good starting model to go into refinement and interactive model building to fix these final things. So here is a detail of the structure where we see how this how the structure evolves during this iterative procedure. So here we have the density and the initial AlphaFold prediction. That was not quite right everywhere. So we see here, okay, we are in the density. Here we're clearly not. The next step, we have this go through this iterative procedure. We rebuild our model according to density. So now it fits better. And now we do our new AlphaFold prediction. So we use this model as a template and get this prediction. And that fits the model quite well. So here, for example, we have this kind of odd angle in our working model for some reason. But in the prediction, this is fixed. And then we go through, this is the second cycle of rebuilding. Okay, that looks quite good. And then we have the third cycle of AlphaFold prediction. And we go to the next cycle. And here finally we have in pinkish, we have the deposited structure. And in green, we have the result of this iterative procedure. That is, by the way, fully automatic. So you don't have to, let's say, yourself going over and over through all these procedures. And so here the idea is really to rethink a little bit this workflow. So here's again these boxes with the different steps in cryo-EM. So instead of doing map improvement, docking, model building, and then going further, we can insert here model prediction and do this several times. And then after that, ideally, we have a model that is as complete as possible as it can be done with current tools. And then we would need some more manual work, or let's say interactive work. And but ideally, this helps getting something very complete. So here are the tools that you can use for AlphaFold models in Phenix. So you can do AlphaFold prediction with Phenix. You don't need to have AlphaFold installed. We have a server where you can send your job to. There's this button here in the upper right. There is a status, so you can see if the server is currently very busy, and then you can decide if you want to start your job or not. You can also let your job wait. There's this process predicted model tool where you can supply a predicted model and also the PAE metrics in this optional input files. And this will do all this processing automatically. You don't have to go through any editing stages yourself. And finally, this iterative procedure, you can supply a sequence and your experimental data, such as a cryo-EM map, and then it will do this iterative procedure for you. And at the end, this is hopefully the most complete model possible. So again, you can ask for several cycles. If you, let's say, ask for 10 cycles, it will stop maybe whenever it sees that the prediction and the working model are so similar that it's not worth going through another round. So even if you ask for 100 cycles, it will probably not do 100 cycles. And again, you can do this on the server, at least the AlphaFold prediction. And so it's 11, so let me go really quick here. The strategy for structure determination now with these predicted structures is the following. The first maybe is to predict your structure and then to design your experiment accordingly. You can use the information of the hypothesis of how the structure looks like to choose your experimental approach. Maybe consider trimming and at domain boundaries for the actual experiment. Then you solve your structure. In cryo-EM, you would do this with docking. And then you update your prediction. So you run AlphaFold again with your best working model as a template. And then you further improve your structure, and then you can iterate this approach. Okay, so I will go over this summary here. Here's some further reading. We have a lot of documentation on our Phenix online website. In particular, there is a one documentation page that lists all the possibilities for using predicted models. We have video tutorials that explain our Phenix tools. And also there are some lectures, and I want to highlight Tom Terwilliger's lecture about AlphaFold. There is a talk from the IOCR meeting. I believe that was a plenary lecture. So if you want to hear Tom Terwilliger's presentation, so Tom did develop a lot of these tools that are presented today, I suggest you go on our website. And finally, I work with Phenix, and Phenix is a collaboration between different groups. As I mentioned, I work at Lawrence Berkeley National Lab, and our collaborators are Tom Terwilliger from Los Alamos, Randy Reed from Cambridge in the UK, and the Richardson Group from Duke University. And that's all for today.